Hello guys, I am Shubham from Tutorials. This video is about endoplasmic reticulum. We will study the organization of endoplasmic reticulum. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. This is absolutely free from our side, but it will help us grow. So endoplasmic reticulum is basically network of tubules and flattened sacs. Okay, I will write here network of tubules and flattened sacs. एंड जो टोटल मेम्ब्रेन है, ओके, टोटल मेम्ब्रेन ऑफ एनिमल सेल का मोर देन हाफ इज अ ईआर मेम्ब्रेन. गॉट इट? ईआर मेम्ब्रेन. तो जो ईआर की मेम्ब्रेन है दैट इज हाफ of the total membrane, more than half of the total membrane of animal cell. Got it? So let's see now. So basically जो ER है that is connected to outer nuclear envelope. Okay, you can see here ये outer nuclear envelope है which is connected to ER. Okay, so if I draw, see, this is a nuclear pore. Okay, nuclear pore, and this nuclear pore outer envelope is connected to ER like this. This is ER. Okay, so nuclear outer membrane is connected to ER lumen, right? Membrane bound है, so that is continuous, basically continuous structure. It is written here. I will highlight the outer nuclear membrane is continuous with the endoplasmic reticulum. Got it? Then the next things we will understand with the help of pictures, right? Important points क्या है? Everything I will be telling you. Now this is showing a fluorescence micrograph of endoplasmic reticulum, right? You can see this outer nuclear envelope area. See this, ये outer nuclear membrane है, and this is ER sheet right here, and ER tubules. These are connected basically. एक network है, right? So it is written here. An animal cell in tissue culture that was genetically engineered to express an ER membrane protein fused to fluorescent protein, right? Fusion किया है so that fluorescence हमें दिखे. The ER extends as a network of tubules and sheets. Network है of tubules and sheets throughout the entire cytosol, so that all regions of the cytosol are close to some portions of the ER membrane. The outer nuclear membrane, which is continuous with the ER, is also stained. Right? Stain green color का हमें दिख रहा है यहाँ पे. जो part B picture है, that is ER network in a living plant cell. See this, plant cell में, right? And this is in animal cell. A picture that was genetically engineered to express a fluorescent protein in the ER. Same, right? So यहाँ पे हमें थोड़ा सा डिफरेंट दिख रहा है स्ट्रक्चर, बट सेम है, नेटवर्क है ऑफ ट्यूबुल्स एंड sacs. Then this ER is basically of two types. Okay, two types, rough ER and smooth ER. Okay, why this is called rough? Because of ribosomes presence, right? राइबोसोम प्रेजेंट होता है, So that gives a rough texture to the ER because of a ribosome presence, and this is smooth. Why smooth? Because no ribosomes. Okay, no ribosomes. Got it? So ये बेसिक डिफरेंस है. जो रफ ER है जिसमें राइबोसोम्स है, तो इसका काम क्या होगा? प्रोटीन सिंथेसिस, राइट? प्रोटीन सिंथेसिस. So rough ER is involved in protein synthesis. Okay, and जो smooth ER है that is involved in lipid synthesis. Okay, I will write here lipid synthesis, lipid or fat synthesis. Got it? So this is a difference. Then see, this is very logical. फिर जो ऐसे सेल है जिनका काम है सिक्रीशन करना, राइट, मोर ऑफ द प्रोटीन्स बनाना, उसमें रफ ईआर का अमाउंट हाई होगा. ओके, एंड ऐसे सेल जो फैट सिंथेसिस करते हैं, हार्मोन सिंथेसिस करते हैं, ओके, लाइक स्टीरॉयड हार्मोन, उसमें स्मूथ ईआर अबंडेंस में होगा, राइट? सो दिस इज बेसिकली डिपेंडेंट ऑन सेल. तो इसमें यही लिखा है सब, लाइक pancreatic exocrine cell that makes and secretes large amount of digestive enzyme every day, right? pancreas का काम है enzymes बनाना in large amounts, तो rough ER उसमें high amounts में present होगा. Okay, the cytosol is filled with closely packed sheets of ER membrane that are studded with ribosomes, right? ribosome वाला कंटेंट हमें ज्यादा मिलेगा. At the top left is a portion of nucleus and its nuclear envelope. From that, the outer nuclear envelope, which is continuous with the ER, is also studded with ribosomes. Okay, so both the membranes are studded with ribosomes, flooded with ribosomes. Okay, जो न्यूक्लियर मेम्ब्रेन है एज वेल एज ER मेम्ब्रेन है, दोनों में राइबोसोम कंटेंट प्रेजेंट है. दैट इज ओनली द मीनिंग. Now this B picture is abundant smooth ER in a cell that secretes steroid hormone. Okay, steroid hormone जो बना रहा है. So the electron micrograph is of a testosterone secreting Leydig cell in the human testes. Okay, so इसमें smooth ER हमें more मिलेगा, ज़्यादा मिलेगा, right? Smooth ER present in high amount in testosterone secreting Leydig cell and rough ER in high amounts in pancreatic cell where enzymes are produced. And the C picture is basically three-dimensional reconstruction of a region of smooth ER and rough ER in a liver cell. Okay, the rough ER forms oriented stacks of flattened cisternae, each having a luminal space 20 to 30 nanometer wide. The smooth ER membrane is connected to this cisternae and forms a fine network of tubules 30 to 60 nanometer in diameter. The ER lumen is colored green. Okay, means see, ये liver cell है, liver cell में rough ER भी present है and smooth ER भी present. Got it? So basically what happens, I will tell you. Let's take a plain slide. So detoxification ये भी एक रोल है, राइट? ER का detoxification of drugs and harmful products, राइट? तो इसमें जो ER मेम्ब्रेन है contains enzymes, basically family of cytochrome P450 family enzymes. Okay, so these are basically for detoxification. Right. So क्या करते हैं ये enzymes? They convert products from water insoluble to water soluble. Okay, I will write here. Water insoluble accumulate हो जाएंगे, right? If they are not metabolized properly. So this water insoluble substances are converted to water soluble compounds, right? And this is how detoxification is done of the harmful products or drugs to clear out from the body. Body से clear करने के लिए ये process होता है detoxification. One more role of the ER is sequester calcium 2 plus. Okay, calcium 2 plus का uptake, release and storage होता है ER में. Okay, storage in ER lumen. So basically about structure अगर हम बोले, see this is a ER. Okay, अभी ये जो line है ना, this is membrane, ER membrane, right? This is ER membrane. Okay, and ये जो अंदर का space है, that is ER lumen. Okay, ER lumen. This is the structure of ER. And to talk about specific ER, okay, specific ER which is present in muscle cells, sarcoplasmic reticulum. Okay, muscle cells have a this special type of ER which is having calcium storage and release function for basically muscle contraction and relaxation, right? मतलब जब muscle contraction and relaxation होता है, myofibril on the tissue level we say myofibril, जब उसका contraction relaxation होता है तो कैल्शियम का प्रॉपर सिग्नलिंग वहाँ पे चलता है. ओके, सो दिस इज अ स्पेशल टाइप ऑफ ER present in muscle cells, right? In muscle cells. So this is also very important, you have to remember. Then this ER makes close contacts with other membranes, right, of mitochondria and plasma membrane. Okay, contact sites बनाता है. So these are known as organelle contact sites between ER and other membrane. So this one membrane makes contact with other organelle membranes also, known as contact sites. This is for the efficient transport of substances like protein and lipids. Okay, protein and lipid का efficient transfer के लिए important है contact sites. Now this ER can be isolated basically and studied in vitro. Okay, in vitro purpose के लिए हम isolate कर सकते हैं by a technique equilibrium density centrifugation. Okay, why so? Because we know that rough ER जिसमें राइबोसोम प्रेजेंट होगा, इसकी डेंसिटी ज्यादा होगी कंपेयर टू स्मूथ ईआर, राइट। स्मूथ ईआर की डेंसिटी कम होगी। So on the basis of this difference, density is what basically weight, right? इसका weight ज्यादा होगा because of ribosome presence and इसका कम होगा. So on the principle of this, we can use a density gradient. So here sucrose is used basically as a density gradient, right? Increasing sucrose concentration. Got it? Then this जो ER है, तो homogenation होने के बाद these are converted to microsomes. Okay, microsomes. Basically, these are small vesicle, right, having the composition of that organelle. Suppose rough microsome, rough ER से derive होगा, right? Rough microsome derived from rough ER and smooth microsomes derived from smooth ER, then Golgi and other organelles also. Okay, so here basically there is a density gradient of sucrose concentration. अभी नीचे जो settle होगा because of density, high density will be rough microsomes. Okay, see, a rough microsome have a high density and stop sedimenting at high sucrose concentration. सुक्रोज नीचे ज्यादा होगा तो नीचे हमें मिलेगा रफ माइक्रोसोम्स एंड ऊपर ऊपर हमें स्मूथ माइक्रोसोम्स मिलेंगे हैविंग अ लो डेंसिटी, stop sedimenting at low sucrose concentration. Got it? Let's read. When an intact cell or tissue is homogenized, many of its membrane enclosed compartments form small steel vesicles called microsomes. Okay, मतलब ये जो compartments है membrane enclosed small vesicles form करते हैं जिसको हम microsomes बोलेंगे. When this mixture of vesicles is sedimented to equilibrium through a gradient of sucrose, two types of microsome separate from each other on the basis of their different densities, right? Densities के हिसाब से two microsomes will be separated. Note that smooth fraction contains non-ER derived material. Okay, very important. जो smooth microsomes है non-ER derived material है, right? Smooth microsomes. Thin section electron micrographs of the purified smooth and rough microsome fraction show an abundance of ribosome studded vesicles in the rough microsome. Yes, जो rough microsome है उसमें राइबोसोम मिलेंगे that originated from the rough ER. These are not seen in the smooth ribosome fraction, which primarily contains ribosome free vesicles originating from smooth ER, Golgi cisternae and other organelles. Right, मतलब rough ER gives rise to rough microsomes. This is important point. And smooth ER gives rise to smooth microsomes. तो है ही, but also these are formed from other structures like Golgi complex, Golgi complex compounds भी smooth microsome form करेंगे as well as plasma membrane as well. This sentence is very important. I will highlight that again. Note that the smooth fraction contains non-ER derived material. तो smooth fraction में non-ER derived material है because that smooth microsomes are basically ribosome free vesicles originating from smooth ER, Golgi cisternae and other organelles. Other organelles भी हैं, okay, but rough microsome is basically rough ER only. Rough microsome fraction that originated from rough ER. Okay, I have highlighted the things. You can refer that. और एक important यहाँ पे study है experimental, right? Secretory proteins enter the ER lumen. Okay, what is written here? See, I will tell you. ये ER है. You can see rough ER where ribosomes are attached and protein synthesis चल रही है यहाँ पे. Right? So these are the proteins. You can see this proteins है. अभी होमोजेनेशन करने के बाद यहाँ पे माइक्रोसोम्स है विच स्टडेड राइबोसोम्स, राइट. अभी एक साइड यहाँ पे क्या किया, ट्रीट विद डिटर्जेंट. ओके, ट्रीट विद डिटर्जेंट. अभी हमें पता है डिटर्जेंट डिसरप्ट द मेम्ब्रेन, राइट. ऑर्गेनल्स की जो मेम्ब्रेन है उसको डिसरप्ट करता है डिटर्जेंट, राइट. सो व्हेन डिटर्जेंट इज ट्रीटेड, membrane is disrupted and then add protease, then protease will digest the secretory protein. अंदर जाएगी ना protease because detergent is used and it has, detergent ने membrane को disrupt किया है, खराब किया है, तो proteins हमारी digest हो जाएंगी. In this case where only protease is used, right, सिर्फ प्रोटीज यूज किया है यहाँ पे जो प्रोटीन्स है इंटैक्ट होगी, कुछ भी नहीं होगा प्रोटीन को, राइट. नो डाइजेशन ऑफ द सिक्रेटरी प्रोटीन. व्हाई? बिकॉज मेम्ब्रेन इसकी इंटैक्ट है, मेम्ब्रेन इसकी डैमेज नहीं हुई है, मेम्ब्रेन इंटैक्ट है बिकॉज नो डिटर्जेंट. ओके, दिस यू हैव टू रिमेंबर. दैट्स इट. लेट्स रीड. Labeling experiments demonstrated that secretory protein are localized to the ER lumen shortly after synthesis, right. secretory protein मतलब co-translational pathway है ये, synthesis के बाद ER lumen में हमें मिलते हैं secretory proteins. Cells are incubated for brief time with radio-labeled amino acids so that only new synthesized protein become labeled. मतलब जो new protein synthesis हो रही है उसको ही लेबल करेंगे, राइट. ओनली न्यू प्रोटीन्स आर लेबल्ड. द सेल्स आर देन होमोजेनाइज. आफ्टर लेबलिंग होमोजेनाइजेशन करेंगे. सेम जो डेंसिटी ग्रेडियंट है ना, सेम जो हमने पढ़ा अभी. फ्रैक्चरिंग द प्लाज्मा मेम्ब्रेन, टीयरिंग द रफ ER into small vesicle called microsome. Then because they have bound ribosomes, microsomes have a much greater buoyant density than other membranous organelles and can be separated from them by a combination of differential and sucrose density gradient centrifugation. जो हमने देखा अभी जो purified microsomes है are treated with protease. Okay, here we are talking of only rough microsomes. Do note, सिर्फ rough microsomes के बारे में हम लोग बोल रहे हैं. Having the role in protein synthesis, right. So when these are treated with protease in the presence or absence of a detergent, the labeled secretory proteins associated with the microsomes are digested by the protease. कब digestion होगा protease से? Only if the microsomal membrane is first destroyed by treatment with detergent. ओके, मतलब जब डिटर्जेंट यूज करेंगे तभी प्रोटीन डाइजेस्ट होगी. रीजन, माइक्रोसोम की मेम्ब्रेन डिटर्जेंट से डिसरप्ट होगी. ये डिस्ट्रॉय होगी। दिस फाइंडिंग इंडिकेट्स दैट न्यूली मेड प्रोटीन्स आर इनसाइड द माइक्रोसोम्स। ओके, क्या समझा इससे? जो प्रोटीन है वो माइक्रोसोम के अंदर है, इक्विवेलेंट टू द ल्यूमेन ऑफ द रफ ईयर। So this explains the role of ER lumen, right? ER lumen में भी ऐसे ही secretory proteins रहते हैं, right? During the synthesis and transport. Okay, so these things are very important of the ER, right? Repetitions में use questions भी आए हैं इसके in the exams. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to our channel, and stay tuned for the upcoming videos. Thank you.